Samantha Belford, and this is my science fair project. My science fair project title is The Science of Luciferin. The testable question that my science fair project is based around is, if I change the bacteria spread's genes, will it affect the E. coli bacteria? My hypothesis around this is, I think the different animal genes will affect my E. coli bacteria by giving it different properties, and I expect the properties will vary based off of the DNA splice I inject. For example, the jellyfish gene will make my E. coli glow because jelly DNA contains a concentrated chemical substance called luciferin that contains properties that can make jellyfish and fireflies glow. At this point, you must be wondering, what is luciferin? An organic substance present in luminescent organisms, such as fireflies, that produces light when oxidized by the action of the enzyme luciferase. The host organism must possess luciferin, a molecule that reacts with oxygen to create light. Did you know that you can actually alter a living creature's DNA and give them new traits? Well, you do now. It's all done with genetic engineering, the deliberate modification of the characteristics of an organism by manipulating its genetic material. My process for this experiment was, first, we need petri dishes in order to keep a sterile work environment. It's really important to keep a sterile work environment in order to prevent contamination for the bacteria, which can ruin your experiment, or to contaminate yourself when you're working with dangerous chemicals or bacteria. Not that the genic E. coli is not that dangerous, but it's a good skill to practice. Anyway, so next we took centrifuged water and spread it on. This is to contribute to the petri dishes being sterile. We spread it on, and we also took the E. coli bacteria that I had. Remember, with E. coli bacteria, you gotta preserve it for as long as you can, so you have to freeze upon arrival. So we let that sit for 24 hours, and then we took it out and we put on the GCF protein. This protein carried luciferin, the stuff in jellyfish genes that's supposed to give my experiment glowing properties. Well, we let that sit for another 24 hours, and then we shone it under a blue light to get this outcome. As you can see, there are white spots, which are very small and hard to see, but it is the bacteria we grown. So this experiment was a success. Here's the data table I filled out, logging the progress of the experiment. Bacteria is incubating on the plates I created. It is starting to develop clear white spots, which means the bacteria is growing. It has been growing for 18 hours and so far is not visible. Next, I just had to put the GCF protein on the bacteria in hopes that we will start to see some visibility soon. The plates have gotten a more yellow hue. This could be due to growth or contamination. I am starting to see clear white dots in the petri dish. 4/29. After letting the bacteria chill in the fridge for 18 hours, we put it under the blue light to see if it inherited glowing properties from the GCP protein. After putting the bacteria under the blue light, we saw glowing white dots in the middle of the dish. This shows that my experiment was a success. My hypothesis was supported. The end product was the E. coli being visible under a blue light. My testable question was, if I change the bacteria spread's genes, will it affect the E. coli bacteria? From this experiment, I learned the process of genetic engineering, safely working with chemicals and bacteria, and about how genes carry traits in your genome, and how that can affect certain organisms. This experiment is a great and early intro to genetic engineering that I would highly recommend. Thank you for listening to my science experiment, and I had a really fun time doing it, and I recommend you do it too.